on in, a couple more seats. Okay, antagonists, protagonists. Take the positions. You like it over here? We're good. Okay, we're warming up. These guys did some push-ups early, so everyone's all pumped up. So, welcome to this year's debate, discussion, fight. Yeah. Drinks can be obtained outside. Beer or vodka, I recommend. So the topic that our colleagues are debating today is undeniably very hot. So the title, unsurprisingly, Connectomics, Missing Link for Understanding Neural Computation or Minutiae of Implementation. Nasty.
That's not our title, by the way.
That's your title.
It's yours.
So now, why should we care? Well, if we just peruse the interweb, as you cannot right now. If you look at Google Scholar, stop. Here's one more great seat right here. It's really great. It's the best. Between 2000 and 2005, Google Scholar told us about it 29 times. Then it went up to 190 times, and then 4,900. And if you look at PubMed, good paper by the way, that one, 32, and then 1,500. So people seem to care, and the question is, do we care, and should we care? Our funding agencies care. So the NIH Human Connectome Project, for instance, has the conceit that altogether, the Human Connectome Project will lead to major advances in our understanding of what makes us uniquely human, and will set the stage for future studies of abnormal brain circuits in many neurological and psychiatric disorders. So we all care, and you should be primed, of course, for the topic, because Brian Wandel, in his masterful lecture for the George Miller Prize, already talked a lot about tractography and its relevance and importance. So we're ready for the topic, and we love it. Now, here are the Hector and Achilles of this evening. In one corner, in this corner here, Moritz Helmstaedter. He is the director of the Department of Connectomics of the Max Planck Institute in Frankfurt. And he is especially good citizen to be here because he was here last week for a completely different symposium. And he just had a small baby. Congratulations.
Thank
you. Hooray. And despite these stresses and hardships, he showed up. Tony Movshon is the director of the Center for Neuroscience at NYU. And Tony, too, had hardships. He had to come up here from below 14th Street. And so, which normally we wouldn't do. But thank you for showing up. Now, we are filming this debate because, you're going to want to have this marked forever, obviously. But I do encourage those of you sitting in the front rows with functioning smartphones, record, take pictures, do everything you can. If you want, you can send it to me. I'm the moderator. I'm David Poeppel. I have an email down there. I'm the right moderator because I have long arms and because I'm colleagues with Moritz at the Max Planck Society and Tony at NYU. And it's a pleasure to be here, and I hope I can stay in the middle and do okay. So we, of course, expect not just a friendly picnic and love fest. We expect vigor and great argumentation because we're near Trump Tower. So the theories are going to be great. The data will be great. And they have some of the best words they're using. So only the best words. And these guys are vigorous because they are muscular in their argumentation. So we have Moritz with an orgy of data and the best possible techniques, and then we have Tony coming in as slicing and dicing. So here's how we're going to proceed. This is the format of the next 4 hours. There will be a brief statement. Moritz will start very brief, a minute or two, followed by Tony for a minute or two. Then each of them will give a brief presentation about the main ideas that they care about in the work, followed by a discussion and debate between the two of them directly. And then we'll open it to the floor and have fun. You guys can bring in drinks. We'll laugh, we'll cry. It's going to be wonderful. So those of you, so there's still good floor seating here. There's a couple more seats up here. So come on in. There's seats there, 2 more here, 2 more here. So come, you're not going to want to walk in later, especially not through the middle because the cameras are. Come on in, come on in, come on. It's okay. Don't be shy. 1, 2, 3, 4. There's one over there. And another one here. Another. Look, there's seats. There's one over there. Come on in, guys.
Over here.
Because since we're filming along here, we want to avoid the.
There's a seat up here.
One more here. Another one over here.
Right here.
You get that one. You get that one. Okay, are we good? Are we ready to go? Is everybody pumped? It's going to be great. Okay, so Moritz, welcome. Look forward to hearing from you.
Ladies and gentlemen, I think this debate will be very short. Just as short as the little worm C. elegans, 1 millimeter. Because that's the one creature we have a complete connectome of. Beautiful. 302 neurons, a little more than 7,000 chemical synapses, all mapped out in detail, precisely. Exactly what any connectomicist would love. We have it. But do we have any model, any complete model of the behavior of C. elegans? Where is it? Of its movement, its feeding, where is it? Tiny, short debate, short worm, short debate. That's what connectomics is.
We could start drinking now. But the sad thing about this is that I've had the good fortune in the last couple of weeks to hear Moritz not once but twice, once at half-hour length and once at full length. And I have to say that I've changed my views a little bit. I used to think that structure was incidental to function, but I now know, like a true believer, that knowing the structure is the only true way to understand function. And so while last week when David actually arranged for us to debate this way, I had a different view. Now, Moritz, as you can see, I have to be you in this because I believe that, not necessarily you, but even poor Sebastian Schunk, who sat in that chair before you, at least figuratively speaking, and others who will sit there after you, really have the truth of the matter. I do have one thing to hold against you, which is that you don't have an entirely pure history in this field. You were a physiologist once.
Sorry.
And I think you need to work on, perhaps re-education in the Cambodian sense for fixing that problem. But in the meantime, I think perhaps you'd like to speak at greater length about the virtues of functional modeling and neuroscience theory.
Thank you.
Okay. I'm shocked. I'm appalled. I don't know what to do. I assume this is April 1st. We have to switch. Moritz, give us your spiel.
So here's the real thing. First, connectomes are complete measurements. What would a complete functional measurement even be? Do you want to map all possible sensory inputs that a given system could ever see or experience? And then somebody comes with even another one, and even another one, and the input space is huge. unsampleable in most experiments. In a connectome, in a map, you have a complete picture of, in that case, structural data. Take a map of New York or a more complicated city structurally. In a map, you have all possible paths laid out. You don't have to measure each of them. You can even go back to a map if you have a new idea and do, in that way, future experiments or prepare for future experiments. So, very simply put, connectomes are efficient and embrace the future. Secondly, of course, for any system, there is a sufficient level of detail. Sure, you don't have to measure everything at the finest resolution. But if you do not know which level of detail is sufficient, then it's very dangerous to stop at a two-course stage. It may just be costly and inefficient to go too detailed, but it's dangerous to fatal if you stop too early. Just watching apples fly wouldn't have told you anything about how electrons behave. You had to measure electrons. Or let's take something else. You know, I know Tony and me, we share the love for classical music. So Tony, do you really want to summarize a Beethoven symphony as the average frequency in 200 millisecond time bins? Connectomes may be too detailed, but they don't run the danger of being too coarse. Third and final point, connectomes help you focus on the relevant parts. Where's my slide? Not knowing that there are tires may distract you and get you working on the heating device in the front of a car for your whole career. Something's missing. It looks almost like a complete car, especially at coarse resolution, you wouldn't notice something's missing. Or if you study the ball boy in a tennis match, very interesting behavior, functionally super exciting, many experiments that you can do. If you had the connectome, the complete map of the tennis court, you might have noticed this huge rectangular lawn thing with a fence in the middle and wondered about what's going on there, because that's something that usually the ball boy doesn't fully access. So connectomes help you focus on the relevant parts. They cut off escape path in hypothesis space. You can't argue away anymore. That's why they are so cool. They are complete, maybe too detailed, but never too coarse, and unforgiving. That's why I love them, connectomes.
Well, yeah, no, I'll take my slide in a second. I like Moritz's slide. I like the car thing. That's kind of cute. Because, you know, measuring the structure of every screw and every rivet in the door of the car is a very important way to understand it. But no, let's not go there. Now, I wanted for this audience in particular to make a distinction that we may end up discussing later. The word connectome has become such an inflated presence in our intellectual lives, as David documented, in part because it has spread from its initial meaning. And it exists at multiple scales. And the reason that that's important is that my first principled objection to connectomes is that you need to analyze a system at a scale that is the natural scale for understanding what the system does. And it's a Goldilocks problem. Just for the purposes of energizing this room, I would argue that the microconnectome that Moritz studies is of too fine a scale. It's, you know, the porridge is, what is it, too hot or too cold? I can't remember. It's a long time since I read that story. That the scale of the analysis is mismatched to the function of the system under study, and I'll elaborate that a little bit. Just to throw something out to Brian Wandel, who's safely back in California, I would also argue that the Human Connectome Project, the tractographer's approach to the connectome, is the other end of the Goldilocks problem. It's too coarse and too crude to give you the kind of computational understanding that I think we actually seek for brain function. There is a level, however, in between of what I'll call, for purposes of argument, neuroanatomy. which is actually kind of a useful level at which to think things. So you can't simply condemn all connectomes, right? The second thing you can't do, and just in case somebody tries to cast me in a Luddite mode, you can't argue against getting data. All right? We're scientists. You're not supposed to argue against learning things. Learning things is good. Learning things is what we do. Discovering stuff, right? So I'm all in favor of discovering stuff, but if you want to actually discover stuff at the level that corresponds to an understanding of the system that you seek, then you need to think about scale. And so the first issue is to think about scale. Now the second one is not entirely fair, because I actually know Moritz's thinking on this, but my slide now, David, if you would, shows that there is a long history of trying to infer function from structure. Every couple of centuries somebody comes along. So we have Descartes, of course, famous for many things, but famous in this particular role because he dissected brains, discovered that the structures within the cranium were all bilaterally mirrored and symmetric, except for one. And because he reasoned that his own awareness and consciousness were undivided and single, that one undivided and single structure in the middle of the brain had to be the seat of all experience and awareness. So it's the pineal body. He's drawing the nerves for olfaction and vision, all converging on the pineal to provide the basis for our experience of ourselves. And that was, that didn't work out so well for Descartes. Other things better, that not so much. But then a couple of centuries later comes Gall and his colleagues, Franz Joseph Gall, who reasoned correctly that the brain had organs within it that had specialized functions, and who reasoned also correctly that the size of those organs might have something to do with how adept somebody was at a particular function. Work of people like Mike Merzenich and many others has shown that brain plasticity can have that effect. The problem they had was assuming that these changes in brain shape and organ size would have an effect that you could palpate on the surface of the skull, and that didn't work out so well for those guys either. So now we're in the 21st century, and we have a much more attractive, much less frightening prospect. We have Moritz and his ilk, right? And they offer us these wonderful three-dimensional volumes showing all the synaptic architecture of all the different structures in the brain, and they invite us to see in those structures an understanding of the function of the system. And my contention is that because of the scale mismatch that I mentioned, and because of a broad failure of a match of structure to function in the nervous system, that those seductive pictures are nothing more than that. They are seductive pictures, seductive in their way, in the same way that Gall's pictures of the human skull or Descartes' pictures of the decussation are. Now, the particular challenge that we face in neuroscience is to achieve an understanding that corresponds to our ability to measure things and work things out. So many of us here were trained as psychologists or trained as sort of systems neuroscientists, and we understand that there are profound limits to how much information we can actually learn from measurement about the system we're studying. One of the things that I think is an important aspect of relating the functions that we measure with the structures that subserve them is there has to be a rough match, a commensurate match between the precision of the structures and the precision of the functions. And our ability to measure function is vastly impoverished compared to Moritz's ability to measure structures. And so that mismatch itself means that it's likely that there are going to be many mappings between the structures that you measure in a connectome and the functions that you observe in a physiological or behavioral experiment. Now, there's some nice examples of that from very simple systems. I'm particularly fond of a paper of Yves Marder. Yves Marder works on the stomach gastric ganglion of the crab, which is an 11-neuron circuit which basically provides an oscillation which controls the contractions and relaxations of the crab's gastric system as it feeds. What she showed in a rather elegant simulation study is that for these 11 neurons, and in fact only 5 of them were part of the study, there are million upon millions of connectional parameters which satisfy the known anatomy and also satisfy the known functional measurements of how that system works. And what's interesting about those parameter sets is that they're all very different from one another. So this same network of 11 neurons can be wired up in many different ways to achieve the goal which we believe it to actually have. And the final thing is that the question of what function you can deduce from structure, I think, is a very vexed one. I happen to have grown up as a scientist in the era of David Marr, and those of us who work on vision recall that Marr articulated very clearly the idea that the function of a system, the algorithm it executes, can be distinguished from the underlying hardware in which the algorithm runs. And it not only could be, but should be. And so we can think about a model which runs on a computer or runs in a brain, and you can think about a particular canonical computation. My colleagues David Heeger and Matteo Carandini have nicely shown that the computation, which they call normalization, can be executed by at least a dozen known different circuit motifs in different animals and different species in different parts of the brain. The computation is the same, the implementation is different. And so to learn the details of the implementation seems at best misguided. And so I think I'll stop there. I will make the point, though, that there's a wonderful paper which I commend to all of you by the physicist Philip Warren Anderson, which is a paper he wrote in 1972. It's in Science. It's just called More is Different. And in it he warns physicists, to be fair, not biologists, but he warns physicists of the perils of trying to deduce how things work from a knowledge of elementary laws of physics. And he has a wonderful quote, which I'll end with, which is simply to say, the ability to reduce everything to simple fundamental laws does not imply the ability to start from those laws and reconstruct the universe. So, with that.
Okay. Okay, thank you, Tony. Thank you, Moritz. So now it's time for mano-a-mano wrestling.
Let me start with a conspicuous leave out that you have there on your slide. So when you're talking about the sequence of deducing function from anatomy, where's Cajal? So because I think that's a great example.
I only did odd-numbered centuries.
because clearly Cajal is the example of choosing a level of detail that was informative and making many predictions that we are still amazed by have held true, from the cellular doctrine to actually the direction of signal flow in neurons, to particular implications about the circuits, how they're implemented without being able to measure circuits in the first place. So I think that's, but I see why you left him out. Now, but I want to focus on something else. I'm really amazed, Tony. I mean, not amazed because I know that. You give me the clear impression that for some reason you have some, to me at least, opaque source of superior knowledge about which scale is the right one for the brain. So you have repeatedly said, don't measure too finely, just stay at the required level that you need. I would love to know which level it is, and especially how you know which level it is. Because I would argue the ones you're suggesting are not sufficient, and I don't think you can really prove me wrong.
All right, I'm not going to answer that for a moment. I'm going to start with Beethoven. You invite me to think that what I'm doing is equivalent to taking the average of the spectrum of a Beethoven symphony and playing it continuously for 37 minutes. I believe that was the argument as I understood it.
Well, bind at 200 millisecond intervals.
It does last 37 minutes. My point is simply that knowing the precise vibrational pattern of every string, of every violin, and knowing the way the particular bow strikes the particular string, and the way that the particular spittle of the horn player falls into his instrument, is not, in fact, instrumental to my understanding of the Beethoven Symphony. In fact, what I want is a somewhat broader, neither too general, which is the average over 37 minutes, nor too refined understanding of the nature of the sounds the orchestra makes.
Yeah, but here is exactly the point. So wouldn't you, or maybe you're not, but I would be interested in knowing that there are 32 or whatever, 20-something violins involved, that there is the leaders of the two violin sets, that there's this thing called a conductor. I would find that mechanistically interesting. I suspect that some of your proposals that there are average values that one can use, you've previously said that you don't want to measure every atom in a solid body if you just want to understand its gross function. Of course not. But how do you know what is the right level of detail? Who tells you which level we need, and why do you know that neurons and their connections are not required?
I have an answer. Would you like the answer?
I'm looking forward to it.
Okay. But I fear you won't accept it. People usually don't have trouble hearing me, David. They want the mic further away.
I want to go all the way in.
Yeah, I want to drill down. So I mean, there's a key question here, which is indeed the question of what the right scale is to do neuroscience.
How do you know? But you seem to know. How do you know?
Calm
Tell
yourself,
me.
Moritz. You're young, you're excited.
Yeah.
Relax. You should have had the drink I offered you, but here's some vodka. I know, but I offered him a drink. He said afterwards.
It's vodka.
Whatever, Tony. I'm hoping you're not only delivering drinks, but also an answer to my question.
So, here's the way I approach the answer to the question. Our understanding, and I'll answer this from the perspective of the system I know best, which is the visual system. But you work on somatosensory system or retina, so you also have a familiarity with this. The system I study is one in which we've done a good deal of analysis at the level of single neuron responses. And in the analysis of single neuron responses, what we do is we create models to match our measurements, which roughly speaking account for the cells in a way that you can think of as being the statistical sum, the statistical summary of a large and unknown set of inputs. So we know when we record from a pyramidal cell, which happens to have a complex receptive field in primary visual cortex, that it has 10,000 synaptic inputs. And some of them come from other simple cells, and some come from complex cells, and some come from several different kinds of interneurons, and so on and so forth. But our physiological measurements don't permit us to see into the cell at that level of precision. What we can see is, in terms of, let's say we fit a standard model to this cell, we might be able to learn the values of a couple of hundred parameters that govern the behavior of that cell. And by the way, that would be an extraordinarily precise account of what that cell's function is. And there is no way, I would submit to you, there is no way, given current technology, that we can actually map those parameters in any meaningful way onto a unique anatomical structure. There are many anatomical structures which could give rise to exactly the same response pattern, and I would submit they're all functionally equivalent from the point of view of understanding the circuit within which that neuron lives. Now, you might argue, and you could always argue, that we will get better at measuring things, and we will learn more about the physiology of these cells, and in time, we will be able to measure not a couple of hundred numbers, but 10,000 numbers. And the problem is that there are laws of variability that govern the way neurons work in circuits that actually limit the amount of information you can extract given almost any arbitrary measurement time about what that neuron actually does. So I think there's a real unmeasurable aspect to the physiology of, let's say, a single neuron, since much of your work focuses on the level of the connections of single neurons. I don't think we can ever measure the functional properties of a single neuron at the level that will let us distinguish the model at the precision that your anatomy would give it to us.
I think I'm not sure I understand why the functional and structural level of detail have to be matched, rather than making sure that you know enough to understand mechanistically how the system operates. But let me ask something very precisely. So when you state you know the neuron, you know how it behaves, you know its receptive field, I think is a term you also used. How can you know that it's not the ball boy that you're studying in the tennis game? So how can you be sure that there's not something more meaningful or more relevant to that neuron than the parameter space you've screened? Because there's so much more you could present.
It's a perfectly valid question, but I'm not sure that knowing all of the neurons' connections would help me. In fact, if that was what I was learning, I would be learning the structure of each square in the net that separates the two halves of the tennis court.
No.
I want to step back.
For instance, let's take your example of the complex cell I think you mentioned. So there's a very concrete circuit suggestion how that could be generated, that kind of receptive field, going back to Hubel and Wiesel and all the following work, of course. Now, if in addition to your notion that the main reason for this complex cell to be there is to have a complex cell's receptive field, if that was true and the circuit was there as proposed by Hubel and Wiesel and followers, I think that would be a convincing point. But without that circuit, you could still argue maybe it's something else it's computing.
No, I think you're missing the point. You're missing the distinction between what I'll call a functional model, which is, in effect, a minimal computational statement of all of the behavior of the cell in as much as we...
But you say all behavior. How can you say all behavior if you haven't presented everything possible to that cell?
Well, that was my earlier point. You can't actually, in finite time, present everything.
That's my point. That's why...
So you have to come up with a functional account, which people try to do, and I won't argue with what success. But, I mean, I'll go to your own work, actually. This is a better example. Your own work on the retina. One of the most beautiful pieces of connectomics, by the way. This is, when David proposed this debate, I explained, I didn't actually want to debate Moritz. I want the people hiding behind him, right? Okay, because Moritz, along with Kevin Brigman and Winfried Denk, did a beautiful piece of work 5 years ago, in which they mapped out the circuit that gives rise to what's called the direction-selective retinal ganglion cell, the mouse retina. And this cell has a property in which it responds to targets only moving in one direction across the retina. And it does that because of a circuit which these guys beautifully delineated at the detailed synaptic level. Right? And they worked it out not because they knew, I would submit, that the particular synaptic architecture of one particular ganglion cell matched that ganglion cell's preference. But they measured multiple ganglion cells, and what they showed was that there was a pattern of synaptic interaction within the interplexiform layer of the mouse retina that subserved that property. What they did was they explained a beautiful set of measurements, physiological measurements, that were made by Barlow and Levick 50 years before. And what they did was they explained it, you explained it, not by virtue of knowing connection by connection, pattern by pattern, but understanding instead what the statistical structure of retinal architecture looked like. That starburst amacrine cells of a particular location in the retina connected to the ganglion cells in a particular way. So that's, to me, an example of connectomics from which the only meaning is to be extracted from the statistical properties of the connection. In other words, what I would call the neuroanatomy. And I'll stop for just a second and I'll put a question to you because I can see you. You're leaning forward. It's that thing back.
No,
If you had not known that you were studying the circuit of a directionally selective ganglion cell in the mouse retina, but if you were studying the circuit of some unknown cell in the middle of the basal ganglia, I submit that you would never in a million years have worked out that that was the function of that circuit.
Totally good point. Thanks for all the compliments. Still, your points were a little bit inaccurate. First of all, we did functionally measure in parallel, so that's very fruitful, of course. But and we did analyze each synapse and found something very unexpected, which is that the selectivity of the circuit even operates on single dendrite level, not just on cell level. So that was really a totally unexpected finding, giving rise to many questions about how this could be possibly wired. But my point is, what we did provide is a final answer to how this is implemented and how this is realized in that circuit. And that for a mechanistic person, well, because before, of course, the option that it was the inhibitory side that primarily generated this or conveyed this directional selectivity to the postsynaptic cell had been around. But there had been opposing views stating it would be the excitatory drive that would create it and other details of that. So of course we live in a functional world. Functional measurements are around, and of course it would be a nightmare not to have had these functional measurements. And of course in any new system I'd love any functional measurement. That's not my point. But my point is, without the connectome, we couldn't have ended the hypothesis fight between those alternating models. With the connectome, for this particular computation, we have ended it. And this is exactly what connectomics is about, in my view, to cut down hypothesis space, of course for functional settings, but for interesting ones. And of course it requires that you're interested in the mechanistics of that circuit. If you don't care about how it's wired up, because that's just, I think, implementation, as you said, then of course, fine. Then you can try to go with some abstract models. I care about the way it's implemented, also because it may be differently done in different species, also because it just tells me something about how actually brains do what you can do abstractly in many ways. So if you care about it, and if you want your hypothesis space to shrink rather than increase over time, you need connectomes.
Okay, I do have to correct one point. I know you want to probably roll this out. The evidence that the connection that gave direction selectivity to the direction selective ganglion cell was inhibitory was overwhelming on the basis of physiology before you did your experiments. Overwhelming.
Well, there was a strong piece of literature claiming it's the excitatory side.
We can, that's definitely too geeky for this crowd. But so from Barlow and Levick on, the argument had been a veto argument, an inhibition mechanism. And by the way, the idea that an individual dendrite would be the substrate for direction selective interaction is implicitly predicted in Barlow and Levick. Okay, because what they did was this wonderful experiment where they showed that the cell, which was not only selected for the movement of a target crossing the whole receptive field, but also little tiny movements within the receptive field. And there's a paper which I will send you because you'll enjoy it, from 1963 by Barlow, called "Are Bipolar Cells Directionally Selective?" Because that was their incorrect guess about the retinal circuit that gave rise to this. My point, though, is still, I think, rather than sort of worrying about the details of this, first of all, to prove that this was an inhibitory circuit rather than an excitatory one, you guys did an awful lot of work that you probably didn't need to do just to make that one point. That's item number one. And item number two, I'd still like to know whether you think you could have deduced the function of that circuit if you hadn't known in advance what it was for.
So for the latter one first, the circuit is wired discretely for four cardinal direction or two orientations and the both signs of it. So we would have found that it's four types of wirings. So would that have told us its orientation? I mean, that would have been a likely speculation in a visual system like that on a flat retina. But the four, so again, the four classes would have shown up. Of course, the notion that its direction would have had to be, I guess. But I would always say, of course, structure can make suggestions about function, but they are meaningful and they allow helpful functional experiments. If you turn it around and just do first an enormous load of functional experiments, by the way, measuring all of that functionally is also a lot of work, and I'm not sure it has been as conclusive. Basing your functional experiments on structural work, I think, is more fun and more conclusive. But never the one without the other. I don't think that's the point.
I do want to agree with you on one important point, which I think everybody should take away, which is that the business of making science work is the business of refining potential hypothesis spaces. And your argument is that connectomics provides an important constraint on the kinds of hypotheses that you can entertain.
Yes.
I would agree with that. I don't think it's a terribly efficient way of doing it. I think if you're going to invest resources in doing it, there are probably better ways.
Okay, no, but this one I really don't buy from you, Tony, because functional experiments are so, are so stressful.
20th century, I know. It's,
They are so, well, no, that's not the point. The point, they are so, exhausting, and they take so long, and then it's just one exposure cycle of whatever your experiment can last, and then.
How many hours were in the material you presented?
20,000 work hours. That's 10 years of a, not so diligent scientist. It's maybe 1 or maybe 5 years of a more diligent one. So I mean, that's compared to how many graduate students do single-cell experiments in your lab and expose a certain type of stimuli, a very small set of functional stimuli, to maybe many neurons, but, that's tedious. I'm impressed by that kind of work, and I don't envy you for having to do that, Tony. It's tough stuff. So don't tell me it's efficient.
I'm touched, but the sympathy is oozing out of here. To me, it's much easier. Neuroscience has thrived as basically by being a cottage industry, right? Hubel and Wiesel ran a cottage industry. Vernon Mountcastle ran a cottage industry. Bert Sackmann, your mentor, ran a cottage industry. One of the other things that I find disturbing about connectomics is that there's a sort of corporate flavor to it. In order to be a connectomicist, you have to be part of a conglomerate. I'm being facetious, but of course, the demands of connectomics, the technical and manpower demands, are enormous. I think
but is this really...
So, I don't think you can really, with a straight face, argue that physiology is harder than connectomics. It's only harder if you don't happen to have an army behind you.
It's maybe harder for more cottages than one a little bit larger cottage. Maybe that's true. But let me here use the analogy.
General Motors a large cottage?
I'm sorry?
No.
Okay. So, but this, this really gets into a science, I guess, policy or even resource allocation question, which I find a little less exciting.
I'm actually not interested in that. So
exactly. So that's not a very now. There's another example. That at some point in time, it's always been cited in the connectomics debates. There was the genome project, and that had a very much corporate style attitude to it in the beginning. Now we're 1 billion times faster, roughly, after 40 years of work on sequencing technology, about a factor of 1 billion faster and cheaper. So now it's everybody's technology, and people are pretty happy to be able to use it and sequence many genomes individually, many species that you're interested in, and so on.
You're perhaps too young, but were you around at the beginning of the genome project?
No, of the 90s genome project. Yes.
Do you remember what we were promised from the genome project?
No.
it would be worthwhile to go back and look.
Well, wait, okay, wait,
So,
okay.
so no, I'm going to...
No, but it's not a question whether each promise was matched. The question is, was it worth it? Do you think, would you prefer Venture to have worked on creating organisms first or something else?
I'm going to go with the space program wasn't a good idea because of Teflon. Okay, the space program was a good idea because we put men on the moon. And the Genome Project was going to provide us with the understanding of human genetic diseases. That was the motivation for it. And it was going to let us understand and then cure human genetic diseases. Now, the Genome Project has had immense benefits for biology in all sorts of ways. It's done many things that were not anticipated by its creators. But it has emphatically failed in its original.
But in a way, it has just propelled science in the right way in this field, also in the medical field. And it's exactly an example of cutting down hypothesis space. Yes, before it started and while it was run, and as a motivation for it, disease curing was put on the table. And the thought was that there would be much more monogenetic or just few genetically caused diseases. The reality is it's not the case, but that's knowledge, that's insight, that's huge because it cut down this bad hypothesis that there are many single gene diseases. It turned to the table that you have this very distributed issue with just probabilities adding up and so on. It's a totally new view of diseases. It has also focused on epigenetic mechanisms again. It has focused on copy number variation studies and so on. So the fact that this wrong hypothesis, which was even, yes, the motivation for it, but fine. It's almost gone now, and now we're ready to find out much more relevant things about these diseases. So would you really prefer that not to have happened just because people were wrong in the reasons to do it, and not even really wrong, just a little bit too optimistic?
The Genome Project, which was a far smaller undertaking than a Connectome project, made some very grand promises which have still not been fulfilled. Understanding that there aren't very many single gene disorders has not actually helped us to do anything much about them. It just means that we know certain kinds of routes will not work.
Let me intervene now because the Genome Project, as fascinating as it is and its problems are quite interesting, is not quite at stake. And so let me ask.
We are making, by the way, much less overwhelming promises. I'm just saying, please let us measure the connectomes, and we'll see.
I'd like to ask something.
You will learn.
Can't measure them.
I'd like to actually ask something of Moritz, and then maybe a question to both of you because of the issue of scales that Tony mentioned. So Moritz, I was going to ask to perhaps respond briefly to Tony's example of Eve Marder's stomatogastric ganglion and the issue of multiple realizability.
Yeah. Two things. If it's just about the issue of multiple circuits could give rise to function, and that's for me just an example of there are many possible implementations we would like to know. I would at least like to know which one is actually used, so measure the implementation. But there's another aspect to this argument, which is look at stomatogastric ganglion, look at C. elegans, the connectome hasn't helped as much. Now, this is actually a very interesting and a little bit difficult argument. The circuits, it may be, and you have good arguments of scale there, that for small systems, counterintuitively, circuits matter relatively less than in larger systems. What do I mean? If you have a system of 302 neurons, you have the possibility to encode exactly each single synapse and maybe even with multiple transmitters and very precisely in your genome. So you can really, over millions of years, create the particular implementation that works and with all nitty-gritty details in addition to just who wires to who, like transmitters, many different co-transmission and so on. In a large system, you just cannot afford that. In a human cortex, there's no way it can be all genetically encoded what's in a single synapse. So you have to, the circuits become relatively more important. It's a difficult point, but it's true.
It's not only difficult, it's rank speculation. You have no basis for it. I mean, I'm not saying that you can genetically...
I give you...
I'm not saying you can genetically specify every connection.
Because that would be wrong.
But that certainly doesn't mean that multiple mechanisms in cortex cannot act to achieve the same goal.
That's true, but wouldn't that be interesting to know if, for instance, mouse has found a different way of doing this than man? I would like to know that. If I finally know how it's working and not just have 10 possible ways and still have to, weigh the one against the other and never know because I need to go back to my functional experiment.
5 neurons give you 11 million different recipes. And now if you imagine a cortical column, even with less diversity neuron for neuron, I still think you have a huge uniqueness problem. And that's the scale problem that David
So let's go to that. And so before we open to the audience, you can get ready for your questions now. Get in line. Now's a good time to prepare your question. I guess I'd like you to briefly to discuss. So all of the discussion has been about really let's say, microconnectomes, or very small scale issues where we have a good grip. But most of us here work on not 5 neurons, but 86 billion. And that presents a different kind of problem. And I wonder if
99 billion are in the cerebellum, right?
yeah, so the 69 billion ones are we don't bother with, and we don't bother with the 1 billion in the brainstem either. But could you just briefly speculate, in the pro and con, in which case, how does this apply to the kind of issues we have to deal with in the type of data on the human brain?
Large scales is definitely something for Tony.
So I would make the argument, and it's based on the same kind of functional modeling argument that I've already rehearsed, so I won't do it yet. There is a project, there are several projects. The Allen Institute is doing one, people at Cold Spring Harbor are doing another, which are concentrating not on the analysis of very small subsections of brain. So Moritz will tell you that the scale on which he can do his analysis is now fairly sharply limited to a small volume of tissue, arguably a volume of tissue which is roughly of the scale that you need to understand a retinal direction selectivity computation, but a challenge for cortex. And he has hopes in the future, and I have no doubt that they'll be realized, that he'll be able to make measurements on larger scales that become more relevant. But there's another approach you can execute now, which is to look at the connections of, let's say, microcircuits of neurons, groups of neurons of the order of scale of 50 to 100 microns, and ask, in effect, point to point throughout the brain, what's the connectivity of small groups of neurons with other small groups of neurons? You sacrifice the ability to know the circuit, each circuit in detail, but you gain this understanding of the long-range actions of the brain. And I'm cheating here because Moritz and I were talking about this morning, but one of the things about cortex, which is the structure that Moritz is now concentrating on, is that much of its activity depends not on local circuits that can be measured with the current microconnectomic approach, but on long-range interactions. We all know this between different areas and between cortex and thalamus and God knows what else. So if you know that you have to understand long-range interactions, at this stage it seems to me that you should be willing to sacrifice the ultimate level of detail in order to get the first order, let's say, impression of what all these signal flows are.
Sure. I mean, some information or important information is in long range, absolutely. We don't know whether even long range can be single cell specific. That would not show up in any bulk measurement, but still bulk measurements are a good starting point, I agree. Combining those with high resolution local measurements is what we're, for instance, going for. I think bridging the gap between microconnectomics, as you can call it, which we are doing, or cellular resolution connectomics, and macroscopic, non-invasively accessible connectomics is very important. People are thinking about it. We're also talking to people. I think this gap is still large. What is usually a voxel in most non-invasive techniques is our entire data set. So
It's your
now,
entire brain. It's a mouse brain fits in a voxel of what most of the folks in the room measure.
well, I was talking about cubic millimeter. I thought you meant my brain.
We're not going to go ad hominem here, Moritz, trust me.
So this, yeah, let's
Okay.
not go American political debate style, because you both used great words. Thank you. Thank you, Tony. So let's open it up to the audience for questions, and you go first.
Okay. I'll just yell.
No, but use the mic's being recorded.
And you should probably tell who you're asking the question to.
This is mostly directed towards you, but anything is fine. In a world where we really have to rely on resource constraints and who gets the money and who doesn't and where it gets funneled, and also in a world where historically so many fascinating questions and answers have come just serendipitously that were not asked for or not hypothesized, what do you think the balance ought to be in terms of putting money into things that you really don't think are going to pay off, like connectomes?
So we more or less agreed that we weren't going to do the resource allocation thing. I mean, you have captured what I would say is a sort of an extension of the cynicism I exposed in my view of how sort of large-scale corporate science works, okay? And it's not just connectomics in general. I actually think that the sciences that are represented in this room and represented by people like me have thrived because of individual initiative, small-scale work, the kinds of things that are thought to be inefficient. This is the cottage industry aspect of neuroscience. So I am concerned about anything, not just connectomics, that calls for a very large allocation of resources that might otherwise be allocated to what NIH would call investigator-initiated project grants, if you want to think about it that way. So I believe that the right scale for having science done well is at the individual investigator scale. I have to say that I'm probably a dinosaur and I'm probably a creature of the past in this regard, because the pressure from the rest of biology, in case you haven't noticed it, is for us to become big and corporate and organized and collective and to think much more about large groups. If you listen to, for example, Christoph Koch, who I'll say used to be one of us and became one of them, right? Christoph, who is the scientific director of the Allen Institute, who has put together 330 people who are all supposed to be working in the service of a single common goal. Okay, it's kind of eerie to have him talk about it. You think, serried ranks. Never mind. But I share the concern, and I'm not sure if it's your own concern, but it's a concern I would echo, assuming you weren't dressing up as somebody else when you asked your question, that this is not the right way to allocate resources. But I wouldn't target connectomes specifically, because I don't have enough knowledge to know that that isn't, that Moritz isn't really right, and I'm going to have to get his badge back and wear it around as a sort of like a badge of humiliation for the next decade. But I'll take your point and I'll agree with it. I suspect Moritz.
Just very briefly, I totally agree with most of what Tony said. Science should be distributed. And really huge conglomerates which really enforce joint action can be very dangerous. But I think we should also here watch the scale a little bit. So it's definitely very dangerous if you're allocating, say, $1 billion to just one thing. That's just a lot of money. But in the intermediate scale of tens of millions, let's say, like the Allen Institute is running on per year or something like that. Well, why not? I mean, also it's private money. So why not? I want to defend Christoph here. I mean, why not let them do their thing? By the way, that's just the anatomy project you just defended comes from that institute. So I don't think it's wrong by first principles unless it gets so big that it really drowns all the other funding. I think science should be distributed, but let us do our things. And again, I mean, we're not large by that comparison at all.
I believe you speak for the German government.
No, but the fact that we're at Max Planck, we have 2 departments, Winfried Deng's and mine, dedicated to connectomics. That's a substantial but not a humongous investment by any comparison. But we're able to still to drive the field.
The argument is any funds that are dedicated to these large collective projects are in principle, and I'm not sure it's true that they are really, but in principle they're funds that could be used in other ways. Now, for reasons of politics and philanthropy and everything else, it may not actually be true, and that's a point I should have also made. Sometimes funds become available because a large project is compelling to funders in a way that smaller projects are not. So there is actually not a zero-sum game going on here, and that's another thing to consider.
You're next.
So my question is...
Quiet.
Is it working or...?
Yeah, it's okay, go.
Lean in.
Lean in. Beyonce, really, right? Put your mouth right there.
So,
Become one with the mic.
So I guess this is a question to both of the speakers. Do you think that the importance and the promise of further connectomics work could be modulated by the particular subsphere of the science? For example, for people who are interested in, let's say, artificial intelligence and neuroinformatics, and kind of trying to understand how the brain works at different levels in order to then implement it in artificial systems. Do you think understanding how things happen at the very little physiological, cellular level is more important for those kind of questions, or do you think it's still more important to explore these things on a higher systems level and a functional level? Thank you.
I think this is a very exciting question, and as you probably know, that it's one that actually gets funded in the States right now by the IARPA with the Migrants Initiative, as it's called, by the way, quite a bit of money. We are pursuing comparable goals with our own interests in my department. So the question is any of the machine learning that has been proposed in computer science that was originally inspired neuronally in a way, but has essentially just had a life of its own. Can we go back to the neuronal implementation and ask whether any of those proposals are actually implemented? And I think or others, right? And only with the recent ability to train recurrent networks and so on, this really has become interesting because you can really match the digital implementation to the actual one in the brain. I think it's a very exciting question, and I think most of the models require a neuronal equivalence. So most of those network models, partly because they were inspired by neurons in the first place. So for those, you will probably need the connectome to map them or to find out whether any of this is being used. I think it's very exciting research.
Yeah, I'm going to take a slight exception to that. I think that the architecture of most neurally inspired models of the kind that the questioner was asking about really have very little to do with particular connectional architectures. They're not really about the connectome. They're about layers interconnected by neuron-like computations. And I think it's also recognized that those computations can be implemented in a variety of different ways that look a lot less neuron-like. Now, that being said, at a slightly more abstract level, I've been in this business long enough to watch sort of AI computational approaches to cognition and vision and other things wander in and out of sync with biology. And I think we're in a period where everything is actually wandering back into sync with biology, and I think that's a good thing. So I think there's a lot to go between those disciplines. It, I think, isn't connectomic, but maybe.
Well, but just to give you a very brief example, deep, what is now called fashionably deep neuronal networks, which is nothing more than multi-layer perceptron. That is one structure. Recurrent neural networks, LSTM trained or whatever, that has now been very successful in sequence learning, is another one. A totally different structure. One highly recurrent to a very high degree with certain tricks as far as now the training goes. The other one, a layered one with whatever other features you can put in between. So this is an example of actually a truly connectomically different even implementation in the CS world. So it's being done differently.
You can't do that. They're anatomically different. They're not connectomically different. You can't have all of neuroanatomy under connectomics. That's not fair.
Okay.
I just wanted to, Dave Everstorff from Missouri. It's flyover country for most of you. I just wanted to thank
Moritz doesn't have to fly over Missouri.
All right, fair enough. Fair enough. But I actually wanted to briefly thank you for not taking up the resource allocation issue, because every one of us in the room needs to be reaching out to the public and to our politicians and giving us a bigger damn pie that we don't have to slice around. Thanks for not taking that up.
So, over here.
So this is a question actually that follows the previous one, which is that the computational models or neural nets that we have today, including the recurrent networks, they really don't directly, I was a neural network modeler myself for many years, but they don't really directly relate to the typology or the anatomic structure of the human brain. So this is how we can link these models to the connectome or connectomics is really unclear. So I was curious as to what Moritz or Tony have to say. I also have a related question, kind of goes back to David's earlier comment, which is that most of the discussion today or the debate today has focused on the microconnectomes. For many of us sitting here, we are really interested in how a larger scale, sort of human brain level connectome works, and whether the microconnectomes you have developed or you guys have looked at really can be informative or can provide some insights and understanding to how human connectomes work. So thank you.
To the first question, just very briefly, even the question whether, say, a local cortical circuit is essentially just maybe even random recurrent neural network, where, as you say, structure doesn't mean more than it's random, or it matches more what has been proposed as this multi-layered, more complex feature computation model that is still around for even the whole visual system, or both of them, or in some combination. I mean, that's we don't know, and we should, but by measuring, we will know. If the statistics of the circuit locally show absolutely no layeredness, no subnet networks, no nothing, then at the end the only remaining conclusion is it's really just that kind of pairwise statistical random model. I should say any measurement we are making in any of these circuits so far has always shown higher specificity than pairwise random, so I think it's unlikely to stay like that, but so far we don't know, so we should measure. Now, briefly, human connectome, of course, ideally, I think we would have Tony doesn't want that, but I would like to have the cellular resolution connectome for the human. That's very difficult because we can't go as invasively, obviously. Linking what we can do non-invasively with what we know in terms of typical circuits locally, I think will be critical. You can think of, better constraint inverse models, better fusion models, and so on, that we could essentially contribute from the microconnectomics side to improve technology that's non-invasive and human.
I'm sure that's true. I would just make one other comment. You in effect asked us to think about the parallel between the architecture of computational models and the architecture of neural structures. And I think that these things are very far away from the kind of connectomics that Moritz in particular is interested in. So I'm thinking of deep networks compared with, for example, the architecture of, let's say, a visual system or an auditory system that does the same kind of classification that the deep network is trained to do. These are very much kind of analogy resemblances. They're not really circuit similarities at any level that's, I think, meaningful for neuroscientists. So I think your question is well taken.
I mean, to give you one example, some of us, for example, work on graph theory. We can apply graph theory and look at the typology of these models applying to different levels. So I was wondering if for something like that, like a graph theory typology would apply equally across different levels of analysis. So, again, I have no idea how microconnectomes work in the C. elegans kind of model.
I mean, people have applied graph theory, or graph theoretical approaches, one should say, to analyze the C. elegans network and other microconnectomic networks. The question is, how informative is it at that level? I mean, finding that there's an input and in the end an output is something you could have guessed without that. So that's currently mostly the level. You can then use other graph descriptors that so far, I think, neuroscientifically have been less informative than one would have hoped. But these are very small networks. So, and the same is true for the retina, very small or very shallow. It's not deep, right? So I think in cortex that will become probably more fruitful.
Hang on, over this side.
So my question is, if, well, for Anthony, but also for both, if we were to redirect our attention to something that is not connectomics, because it's not giving us all the answers it promised, which approach to brain function today would you tell people to look at, or to also look at, like in parallel to connectomics? So the answer is not only in connectomics, but it's also in what? Like given the tools we have today.
I guess that one's for me, huh?
Yeah.
So, it's kind of a cheat, but what I'm going to say is I wouldn't presume to answer that, because what I would tell you to do is to use your own initiative to come up with ideas that you can then use to persuade me that your approach is a good one. That's the nature of individually initiated science. We all come from different backgrounds. We all have different priorities. My priorities are not likely to be the same as yours. And what I'm saying is that I probably shouldn't be the person setting your priorities. You should. And so the answer is it's on you, not on me.
Yeah, but I was not like talking about my priorities, but like in general, like for the general public, if they get the idea of all this popular term of connectomics and it's going to give us all the answers, and you're saying, well, it's not going to give us all the answers. There's also these other approaches you should look at. So what would you tell like somebody that's not doing neuroscience, that has heard that the connectome is the big answer to everything? What would you tell them to look at besides
How about the rest of neuroscience? I understand why you're asking, but I'm really not going to answer the question because no particular answer is right. The answer is, in general, you want to think about the priorities with which you decide on things like funding, and you want to balance them against likely outcomes. And I think Moritz and I would probably have rather similar rankings of priorities of most things, and we would differ here and there. And so, we all want to know everything we possibly can about the brain as quickly as we can. So, and I think the mythical member of the general public, by the way, who doesn't give a crap about any of this, is just between us, is not really going to worry about what we particularly come up with. They're going to like answers, not the particular questions we choose to ask.
Okay, that was not satisfying for her.
I'm
But let's see if you can do.
a long list of people I haven't
a long. Let's. Glad I didn't say that. Now.
Shall I talk about my mother? No.
Let's see if you guys can do better on this side of the room, over there.
Hi. So I have a question about variability. Can you hear me? I have a question about variability, and it's in the scale of variability we're talking about. synaptic or cellular connectomes. So I don't know anything about the scale at which there is variability either within one person in their synaptic connections over time or across people in the synaptic or cellular connections. And if we knew something about that variability, would we find that the variability in synaptic connections in one person over their life or across people is so big that it rules out a microconnectome as being a useful tool because it changes almost like a Schrodinger's box. It changes so quickly that we can never actually pin it down. Or is it at is it at a scale that is small enough that actually measuring a single connectome is useful? And I guess as an analogy, in the human connectome, there's variability in the Human Genome Project, there's variability in genomes across people, but that variability has been characterized in a way which is capturable, that there's a few number of options at any one position in the genome. Do we know that the human connectome has anything like that it's a manageable level of variability?
I think it's a very important question, if I may. It's a very important question, and I think the genome example exactly tells you what you want. You want an answer by measuring. And that's what we're doing right now. So I have two answers to that. In the retina, we know it's well conserved. So the circuits we've found, we've seen in several samples. It's there, and you can work across individuals. They are there. The exact quantification of variability is still outstanding, but it's in principle it's highly conserved. Cortex is an open question, but exactly in your analogy to the genome, we should measure, and then we'll know. And that will help us understand what level of description. That's exactly what I said before. Of course, I think we do not know yet the level of descriptive detail that we need for cortex. And I would like to know by measuring rather than assuming I know the level of detail that is required. And I think that's exactly...
We'll work that out. I would just add, Ed, that I think one way to express what I was trying to say about the right level of measurement is to say the right level of measurement for our first pass understanding is the level at which local variations don't matter. Right?
I'd like to take on that because this is actually my question. It's about cortex. So Tony, you're privileged because you work in V1. We know a lot of stuff about V1, both at the neuroanatomy level.
Sorry, is that because I work there or do I work there? Sorry, I couldn't resist.
You just happen to be lucky.
Yeah, okay.
So
Thanks.
you contributed to doing it, that's for sure. But we know a lot about that area. So we have a canonical model, which actually was worked out in the cat, not in monkey yet, but maybe there's been more work done in monkeys that would match that canonical model. But we don't know much about the rest of cortex. And that's very troubling for those of us who are working further away from primary visual or any sensory cortices. So knowing that, to me, is really important. What's the microcircuit organization? How does it contribute to the flow of information? This is a primordial a question to me and to people in my field. And then when we talk about the microconnectome, I don't see how it's going to inform us of that. It's sort of like, let's go way into more detail. And the issue that I have also with the approach with the microconnectome is that, well, you're dealing with different species. We know that there's huge differences in cortical organization in the mouse versus the monkey versus primates. So how is that going to apply to, answering the question, what makes us humans, for example?
We're measuring it. I mean, that's exactly a question we can measure. So you said you are interested in the microconnectome. We're working on providing it also for higher order cortices, of course. And by the way, starting to find interesting things that are not there in early sensory cortices in terms of local circuit structure. So there is a lot of structure in there as far as we've looked so far. And then species, that's what we're doing. Even mouse to rat, 3 times more neurons in cortex. We don't know, is it exactly the same, just 3 times more neurons or new circuit tricks? We'll measure. Than monkey and primate and human. So let's measure. Let us measure, and we'll provide answers to these questions. There are similarities. There's a step from late in the species axis, so to say, with new tricks or not. Maybe it's all random. I mean, but let's measure, and then we know more. I think that's the strategy from my point of view.
I agree that making more measurements will help. I
No,
just disagree
seriously.
about which particular measurements you should make, because I think by the time you've given us a connectome for V1 and the parietal cortex and the frontal cortex in mice and monkeys and humans and rats, Martin and I will be long retired, if not pushing up daisies. So,
Maybe you.
You too, buddy. The point being, I think that you mentioned the magic words, the canonical microcircuit, which is an idea that Kevin Martin, I think, and Rodney Douglas sort of gave us. And it may or may not be accurate, but the idea that there is a sort of fundamental unit of cortical computation, I think, can be illuminated, actually, by the kind of work that Moritz does, but I don't think you can illuminate it by studying every species in every area. You have to extract the principle and see how the principle applies to different.
But then you can still ask whether each species implements it in the same way or not. That may be interesting.
You can't know that at times.
Well, I hope I have time.
Well, look, in the spirit of keeping both our speakers and the audience alive, let's have the next.
Hello, you hear me? I'm interested in the C. elegans. I was reading an article on AI, and they were talking about the C. elegans, how we achieve the connectome on that. We managed to map that into a very simple robot that could behave like the C. elegans. And they were taking this as an evidence that we are closer to the AI. And so now I come to this debate, and if I understood correctly, you said the C. elegans connectome project was a failure. So I was.
Thanks for asking this question. This was our initial statements. We were trying to switch roles. So usually I have to fight against the argument that C. elegans, stating what you just said, that the C. elegans connectome proves that connectomes don't matter. That's usually what I have to fight, and so we are switching roles. So that's why I said it. Sorry. No, I think it was absolutely no failure. It's very no. It was absolutely relevant for C. elegans research. But what is true is that the complete model is missing. But the reason for that is most people would probably say is rather still lack of knowledge of transmitters and other aspects. So the wiring diagram is very helpful.
Can we nail you down on that? Actually, is that what's missing in the C. elegans story?
I mean, I don't work on C. elegans.
So what I'm interested in is, so there's a deep difference between the two of you of what you consider an explanation. And I guess I'd be interested in what's missing for you from the C. elegans case. Is it more about the implementational level of analysis, like, more detail about transmitters or whatever, or do you actually at some point need a Tony-esque view or a Marian view on the problem? That is, will an emergent explanation appear when we know more?
No, that's not given, not a given, and it takes a lot of thinking and intellectual work to do that one, the Marian step that you mentioned. But even the just mechanistic replication is maybe coming, but most people would say not yet there fully. And that one, most people would argue, is because there's, yes, still lack of knowledge about, for instance, transmitters. That is a big problem there. But again, I'm not working on that system, so I shouldn't go too far there, but that's what people would probably say.
Thank you.
Dennis.
So I'm hearing an awful lot of agreement for a debate. So you're both very smart, very well read, and you speak extremely clearly. But as the debate has progressed, I sense a great convergence, which isn't what I had expected.
I haven't moved. It's him.
And so Tony acknowledges that...
He hasn't noticed that he moved.
Tony acknowledges that data are always good, so if it doesn't cost too much, why not? And Moritz acknowledges that it is appropriate to analyze at the level that matters. He just isn't sure he knows which it is. So I would ask, might each of you tell us what it is that you really disagree about?
Well, I think, Dennis, you're being a little disingenuous, because I think we laid out some points of disagreement and kind of left them lying there, steaming in a pile on the ground. The issue of scale is the one that troubles me most. I think Moritz's argument, if I can put words in his mouth, would be that by starting at the cellular and synaptic scale and working up, you will derive a full and complete, I think he used the words complete, understanding of the circuit containing all the elements whose connections you've decoded, and from that an understanding will emerge. And I don't buy that, because I think the scale at which the analysis is done doesn't match the scale at which you want to achieve the understanding. So I think that would be one significant point of difference between us. You can't argue against collecting data, but I actually think that that collecting data of the right kind, that we've already had this go by several times, is probably more important than just collecting data willy-nilly, because you can. So there's actually, there's a guy I admire a lot, actually, who I read recently, a quote from him in the press, who said, said putting together lots and lots of data does not create new science. That was actually Moritz in Science magazine recently. He was commenting on a paper from the Human Brain Project. But I think that's I think that's a well taken point, and I would certainly embrace it. New data alone don't create new science.
Yeah, so that's true. The quote was actually, if it's not the right kind of data, so if you're missing something important like the tires or the wire, the wheels of the car, then that's kind of bad for understanding how it works. I think the real difference is in the optimism of just functional measurements and knowing the right scale, and that's a real danger, I think, because if you're wrong in your assessment of the scale, we may never get to where we want to get to, because if we're too coarse and stay too coarse, we'll just stay there forever. So I think that's, in my view, the only real disagreement, that you think you know the level of scale and think my level of cellular connectomics is too detailed, but again, I still don't know based on which previous knowledge.
So I'm sorry, Dennis, I'm going to cut you off for a second. There's one ground truth, I think, that I have to put on the table and see if we can agree on this. So there's a famous saying of Theodosius Dobzhansky, who is a great biologist, who said, nothing in biology makes sense except in light of evolution. And somebody else, and I'm not sure, it might have been me or it might have been somebody else, somebody in the room may know the origin, has a comparable quote which says, nothing in neuroscience makes sense except in the light of behavior. And so the answer is that the ground truth, the question of whether the scale is right, whether the explanation is creative, whether the explanation is appropriate, is to be found in whether you can come up with a quantitatively accurate account of behavior. So it's not like I'm wandering around with an intuition that this or that is the right level. The question is, can one account for behavior on the basis of the analysis? And I think you would probably accept also that the ultimate goal is to account for behavior. If that's true, then we do have a benchmark we can share.
Sure, but intermediate steps may be allowed. So it's not like, unless you have an immediate prediction of behavior.
So if I may, so I do think there's a fundamental disagreement between the two of you. That is, the downstream goal appears to be the same, namely, let's say, a complete understanding, whatever that means, by the way. Understanding is a complicated idea. But the notion of how to reverse engineer the brain and its foundation for behavior seems a little different for the two of you. Because the approach to reverse engineering is quite different. It appears to be the case that a circuit level, or let's say a connectomics-based analysis, is going to yield the right algorithmic analysis. And as far as I can tell, Tony absolutely doesn't believe that that's even possible, or it's incoherent even possibly.
Well,
and so the question is, where do you get to the right? So for you, the algorithm would come from a careful analysis of the behavior, and for you, it would come from a careful analysis of the anatomy. And I'm actually not sure these are epistemologically compatible between the two of you. I would like the two of you to agree. I even have an illustration of that. But I don't think you do. So I think that the reverse.
Do we have to? No, we don't. Do we have to do this?
No, you don't have to do this.
No.
These are so, but I'm not sure that there's actually where you're going to meet on this in terms of how to cash out experimental designs, for instance, how you would approach it. Seems not so clear to me. It seems like a more profound disagreement than it's becoming clear.
It's the Marr argument over again. Implementation versus algorithm, really, when you boil it down.
I mean, that's a fair distinction. I think the question for me is only whether you have a chance without knowing the structural data. And again, in the retina, we did not. In cortex, we'll see. That's, I think, the disagreement. I don't think it's automatic. I'm not saying structure will tell you everything you need to know. But without it, I doubt that most of the interesting things can be done.
Can I just make one comment?
You can make a short one.
Yeah, so
Person who's been patiently standing behind you.
My summary of the two statements, which again were very clear about your differences, is that Moritz thinks it's safer to err on the side of going too low in the level of analysis. And Tony feels that if we go too low, we'll miss the boat.
Yes, I prefer not to err at all.
Okay. This will be, unless there's pressing questions, in which case you better come up to the mic right now. This will be the beginning of the end of the session.
Thanks very much. That was a lot of fun. This is for Tony, and pretty much following up on the previous comment. I agree with you. But truth be told, I started as a physiologist too, So I also agree with him. So I guess my question is, and you said, you know, we want to collect data, even if it might be boring. Why just let him do his thing? Yes. Eventually, it could pan out. So, who's he hurting really?
So I really don't want to be mean to Moritz, okay? Even if it were in my power to be mean to Moritz, which it isn't.
Not him.
The only answer to that, which we scrupulously avoided in the main part of the debate, was the argument that all of those euros that are being sucked up in his, you know, armies of undergraduates would be better spent buying me microelectrodes. And you know, that's definitely a debatable point, right? And I'm not going to make that argument. I'm fine with him doing that. I'd prefer him to do it over there, right? But that's just, you know, and then we can come here and talk about music. It's more fun.
You have the you don't want to respond to that one? You agree? Wait, no, Moritz, you don't have to. You're off the hook. You already gave us a good cut. No, you gave us the right one. Closing statements.
Wow. Crap. Do I have a closing statement?
No, you don't. You have to make it up on the fly.
Damn. And quote Moritz again?
I'm sorry, there's not a closing statement yet. There's questions still happening over there.
So I was wondering if we even have a basic agreement or understanding of what is the function of a connection. We assume it's always about transfers of information, but we can think about it also in different functions at the biological level and the functional level.
Tony?
So I think, to me, information is actually transferred only in a trivial sense by every connection, right? I mean, it's patterns of connection linking the activity of patterns of neurons with patterns of activity in other neurons that are really representational of anything. I mean, I've spent all my life studying single neurons, but I know perfectly well that single neurons don't actually tell you much of anything. They only work because they're part of an ensemble. We know a good deal of synaptic physiology. When Moritz used to be one of us, he did, you know, elegant work of the kind that Bert Sackmann has done a great deal of, showing us the kinds of signals that can pass from neuron to neuron. I don't like the term information per se, but I think we have a good understanding of the diversity of signals that can pass. And I think Moritz's point is well taken. In creatures like C. elegans, there may well be multiple signals that pass in a way that don't commonly pass between neurons in our brains. So, you know, I have a feeling I'm circling around your question, but I'm not sure what kind of answer you're looking for, because I sort of think as a physiologist we do know, but I have the sense from your question that you think we don't, and I kind of want you to tell me why. You had all those years at NYU, Daniela. Why didn't you tell me this? Oh, sure. I mean, in other words, so there are questions that are, I mean, I think there are connections that are thought to be modulatory in function, connections that are thought to be sort of feed-forward or feedback information passing. I mean, I think there are a variety of forms of connections, and indeed, I mean, I think one of the challenges that Moritz's analysis has is to understand whether a connection in the sense that, for example, Murray Sherman has argued, is a modulatory or what the other one, the driving connection. Sure, there are many, not all connections are equal, I think is something we can certainly embrace.
Totally, yes.
A terrific and juicy last question.
Okay, so I've been trying to figure out what you guys mean, and
both of us?
It's both of you, and I've come to a conclusion. I just want to know what's wrong with it. So I can measure behavior. I know what behavior is. I know where I start. You know, I can observe behavior. I can measure it as a psychologist or even a physiologist, whatever. But if I'm going from structure up, if I'm going from, you know, this thing called connectomics, where do I start? Where is the bottom? Why do I stop at the level of stuff inside the cell? Why don't I go subatomic to understand what's really happening with you know, certain molecular structures that manage to change and make the cells work? I mean, it seems to me that we don't know what that bottom is, whereas it seems to me we do know where behavior starts. And so I sort of have some sympathy with the idea, you know, let's consider what's necessary in order to model these behaviors within some very, very crude substrate and work down from there. And I'm just trying to figure out if that's really the fundamental difference between the two of you. You know, starting from behavior and working down, or starting from the bottom and working up. But if you're starting from the bottom and working up, where is the bottom? I just wanted to get the word bottom in there, I'm sorry.
And I just wanted to get in that it's turtles all the way down as well.
I think that's exactly the question of required level of detail that we were discussing. I think in most really exciting scientific questions, we don't know a priori which is the right level of detail we need, and that was true in the exciting physics of the last 100 years, and that's true here. I don't think we know, and it's difficult because we have to make an assumption, measure at that scale, and then find out whether it's sufficient to go down or up, depending on what the results tell you. So I don't think there's an a priori argument, again, a little bit different from Tony. I don't think there's an a priori argument which level is right. I think only the behavior down approach is something that neuroscience has been doing for a long time now, and that's fruitful, but I think the other approach has also its merits. Maybe that's the way of putting it. I think, if I may, we have a fascinating structure in our heads, and a unique feature of it are the complexity of the networks in our brains. Whether that's just structurally amazing and nothing else, or at the core of the computations carried out in mammalian brains is something we'll need to learn. But I'm very excited that I think the coming years will help us on this way, and we'll do our part, and we'll hope that Tony and your side of things will also do your part.
With such a sentimental sort of resolution into a C major chord glowing, who can possibly disagree?
Well, thank you very much. Thank you, Moritz. Thank you, Tony. You guys have earned your drink.
Something